Move on to our next talk. Just Gordon Broderick will be talking about harnessing AI to accelerate a rational vaccine design. And with that, I will pass it over to Gordon.
Hi, everybody. Thank you so much. It's such a treat to be sitting with such a distinguished group of speakers. I'm going to share with you some of the things that we are using AI to do in the context of vaccine development. So I'm coming to you from Saskatchewan, Canada, which is smack in the middle of the country. If you go to Montana and drive north, you will drive into Saskatchewan. We are Canada's National Pandemic Research Centre, VIDO, and we are the larger of only two containment level 4 facilities in Canada. So we work with dangerous pathogens. You can think of CDC Atlanta as being similar to our facility. So we are a secure facility. As the name would imply, a big part of our mission is to develop vaccines. Vaccines conceptually are built from two components. The first component is an adjuvant, which is a bit of an irritant to the immune system, which calls all the immune cells to the table, summons them. And the second component is an antigen, and an antigen is essentially a fragment of a viral or a bacterial protein. And together these are presented to the host, and the first thing the host does is recognize that pathogen protein at the molecular level. So a part of our work is conducted at the molecular level, and then we're going to be using AI for that. And the next thing is whether this recognition event at the molecular level actually ripples through the immune system at a broader scale and alerts the immune system and creates an immune memory to that pathogen. Now, this is traditionally done in a very resource-intensive experimental environment. And so the hope here is with our group to start assembling an immune digital twin. So that closely mimics the responses of the animals and human immune systems so that we can run through these different combinations of antigen and adjuvant and arrive at more optimal solutions. Okay, so constructing a digital twin. One thing that is a bit of a challenge at VIDO is because we are a level 4 containment facility, samples can't leave. These are dangerous pathogens, so when we conduct experiments, samples can't leave containment, which means that typically we're in a data sparse or data poor environment, at least experimental numerical data poor. In looking at this, we've expanded our idea of what data is to what other people have collected as data, how they've interpreted that data, and then how they've published this for the community in peer-reviewed publications, and maybe stored some of their insights in databases. So the first part of our process really is to go through what we already know by unleashing search engines for schema, so basically extracting everything we know about biological pathways and their connections and their elements and the proteins that are part of these pathways, and also unleashing natural language processing engines, many different types, to go through essentially our biomedical and medical libraries to pull out everything we think we already understand about the immune system and how these immune cells interact with one another. So once we've recovered these truths, air quote, we are still left with gaps. We know a lot about the immune system, but there are still many things that we don't know about the immune system. And this is where we bring AI into play. We're using AI as a hypothesis generation engine, sort of an educated guess of what some of those missing pathway elements might be. And so once we do that, we push those into the same pathway with the recovered truths. We mesh them together so that any hypothesized pathway element has to play well with the ones that we consider to be true. So it has to work well in the context of the ones that we consider to be true. And it has to help us explain what we're seeing experimentally. So we're using, because we're in a data poor environment, we're using data really to do model selection rather than model construction. Right, so it's really, we're at the opposite end of the big data spectrum almost, if we think about just the experimental data. And of course, once we have reasonable families of models, we can then start looking at how resolving differences between these candidate or these potential explanations of the biology, how resolving conflicts between those explanations might inform us on how to perform the best, most informative, most insightful experiments, right? Because experiments in a level 4 containment facility are very challenging to do. They're not without risk, and they're certainly very expensive to do. So there's a big onus on us to make the very, very best of what we already know, make the best guesses possible, and use those to inform on what are the next best set of experiments to do. So as I mentioned, AI comes in here as a hypothesis generation engine. We have been using different types of AI to answer the question, well, do certain proteins in our biology and proteins that are brought into play by a virus or a bacteria, do they actually connect? Can they connect biochemically? So we have been using AI-like models to do rapid screening and essentially try and predict the results of much more detailed molecular and atomic level simulations that take days and weeks to resolve. So we're trying to accelerate that pipeline by using these AI models to suggest which ones might be the best candidates to actually launch into these large atomic resolution analyses. The second one is if they are able to interact biochemically, what are the functional consequences of that interaction? And so this is what we're going to be talking about specifically today. If I know that two proteins might interact with one another, do they regulate each other positively, negatively, or potentially not at all? Okay, and then we use, of course, this to design maximally informative trials. So the first thing that we did when we started exploring this domain was to ask, well, what kind of model do we need, and how big a model do we need? So we've done quite a bit of work looking at model selection of different types of AI models, and the transformer models seem to work reasonably well for us, better than most other types of models. There are quite a few, again, to choose from, even if you're just looking at transformer models. Some are very big and some can be very small. The thought here is that because we're not interested in, we are, but not specifically today, interested in who Ryan Reynolds is dating at this particular instant, we may not need a very broad a model that has very broad knowledge and can answer a broad range of questions. We really are looking for models that have a very narrow focus but are very good at what they do, don't need a lot of data, and importantly, because we're a secure facility, can actually be stored efficiently and taught rapidly locally. So because we're a secure facility, we can't really store our models, or we're being encouraged to not do that, and I'm using a euphemism here. We're being told to not do that. So our models, our AI models, language models of different types, have to be stored and kept locally. Right. So really, that would push us towards smaller, more task and domain specific models. And here's an example of some of our exploratory results here. This is just straight out of the box results where we're doing a standard 80-20 split. I'm sorry, we have a number of provincial and federal ministers touring the facility today, so there might be a little bit of noise here in the background. But these are preliminary results on some of our language model trials, and we essentially looked at how well, what is the incentive here in going to these very, very large models. So we looked at a transformer, basic transformer model, BERT large, BERT, which has over 300 million parameters, a base model, which has about 100 million parameters, a smaller one at 67, and a very small one at 4 million parameters. So this is really small if we think of the trillions of parameters of the larger GPT-type models. So this is very small. And the question here was, if I give you almost all of my data, 80%, and I validate on 20, how well would you do in a few-shot scenario? And we can see that the smaller models actually perform reasonably well, and there's a bit of a sweet spot here at just enough parameters to improve your performance, and then as soon as you go too big, then things start to there's not very much incentive really in going to a much larger model than around 100 million parameters, and that's pretty much what we've worked with so far. Now I have a model choice. How do I teach it? How do I teach it to be a vaccine development specialist? Well, we took a page here from the folks at DeepSeek, and that made a big splash. It wasn't all that surprising to us because, as many people here I'm sure are well aware, not all data is informative, and introducing large amounts of unreliable and potentially redundant or uninformative data can really affect model performance. It can introduce significant bias, and it's very inefficient. And in particular, in our case, it probably will never happen because it's just too expensive and too difficult to generate. Right. So we're very interested in the aspect of reinforcement learning, and we've worked with a more static version of that, active learning in particular. And as part of our exploration of how to tune and teach these models, we've sort of gone back to grassroots conceptually as, what do we do in terms of model selection? So if I'm looking at a large data set, how do I pick the best, most informative data elements out of that data set? Most of the work, I think, in the literature so far, and again, this is our personal experience, we'll own this, has been that the policies typically involve some measure of uncertainty, of novelty in the next best data element. Now, we have looked at those very carefully. Sample selection like EGL or diversity sampling. But we've also looked at label-aware methods, which we have found to be really interesting. And in cases where you don't actually have the real label, we're now working on models that would predict the probability of us being correct or incorrect. So we're working with two elements here of two selection criteria. One is, how uncertain am I in predicting a result for this new sample? And secondly, how correct do I think I am? So we've been playing with this, and here's, again, some preliminary results as to how these might perform. So what we call our low entropy incorrect is a policy that we've been working with that essentially gives a high priority to selecting a data element where I think I'm very certain of this prediction, but I'm wrong. I think anybody who's walked into a final exam and has been really, really confident of their ability to ace that exam and walked out with a C or a C minus or a D, that leaves an emotional scar. And it was interesting for us to see that some of these language models kind of mimicked that human behavior, that if we gave it data elements where it was uncertain but actually ended up being correct, it didn't have the same staying power, or it didn't give us the same learning rate for the model as if I gave it elements where it thought it was really sure but ended up being incorrect in its prediction. So we're starting out here with only samples of 10, and then we're incrementally adding elements here. And this is to predict, does protein A regulate protein B? And we can see that our label-aware strategy seems to perform reasonably well, and it peaks out at an area under the PRC curve of around 80%, and it does that with about a third of the available data, and in some other problems even less, about a quarter of the available data. So this is really interesting for us to see, because again, we are in a data sparse, data availability constrained environment. Here is just an example of what those PRC and ROC curves might look like. And this is mid-course. And as I keep giving it, even with the same policy, is like if I keep providing additional teaching examples or tuning examples here, performance starts to deteriorate. So even if this is reasonably good data, I start confusing the language model, and there actually is a sweet spot, around mid-course of the training here, where I have a reasonably good area under the PRC curve and likewise under the ROC curve. So how do we know if any of this is good or bad or true or false? Well, the first thing we do, as I mentioned, is that we cast these proposed or hypothesized pathway elements or protein-protein interactions in the context of what we actually do know to be true or consider to be true. Relationships that were extracted from pathway databases that are manually curated, and as a second tier, also text mined from large amounts of scientific peer-reviewed publications. So it has to play well with what we consider to be true. And secondly, it also has to help us explain what little experimental data we have available to us, right? So those are our two sort of criteria, even then we will end up with competing models where in some cases it is used or in some cases it isn't, right? And that's where those experimental designs come into play to sort of resolve or do that tie breaking in situations like that. Okay. In building our digital twins, of course, we want to scale up to larger, more detailed, more granular, and hopefully more lifelike biological regulatory networks that capture these interactions between a viral or bacterial pathogen and the host immune system and its dynamics. So at this point, our group is only about 14 months old here at VIDO. I migrated my lab from Upper New York State, and prior to that, Florida, to come to Canada to start this. So it's a relatively new initiative, but we've already migrated pretty much all of our code from CPU to GPU. And so we are primarily working on GPUs. We're looking now at tensor accelerators like the TPU that Google just released as an opportunity to increase the scale and reverse engineer the regulatory logic on larger and larger networks. We've also been working with quantum computing platforms like the IBM Quantum System 1. This is the Quantum System 2, but this work was done on the Quantum System 1. And this is an example of an African swine fever pathogen host immune interaction network. Right now, we're looking at scaling up to tens of thousands of proteins interacting through in excess of 300,000 to 400,000 relationships. So this can get quite big. And over the past year, we've experimented with moving some of this at least to gate-based quantum computing, and it has shown some promise. We're also looking at annealing architectures on the quantum computing side. And of course, I'm just the talking head here. This is work that's all being pushed forward by a very, very talented team that I'm very grateful to be working with. Joyce Reamer has a mixed background in physiology and biomedical engineering. Pranta Saha, she's essentially our computational biology lead. Pranta Saha is our generative AI and machine learning and natural language processing lead. He's a computer science graduate. And we have, of course, Noel Pham family is complete without its electrical and computer engineers. We have two very bright, young, early career people here at the University of Toronto. So what's interesting, I think, is that we're pulling in people from a broad variety of backgrounds, from biochemistry, microbiology, immunology, math, physics, computer engineering, computer science, even physiology, right? So it does take a village to build these kinds of models successfully. And I just want to thank, in closing, our sponsors at the Canadian federal government and provincial governments, as well as our campus collaborators, our advanced research computing team here at the University of Saskatchewan, and our Center for Quantum Topology and Applications, as well as IBM and PinQ Squared, our partners on the quantum computing side. So that concludes my talk.
Thank you so much, Gordon. I think we have just a minute, just one time for just one quick question, if anyone has a quick one. Otherwise, we're at time for the next talk.
Yeah, I put into the chat whether or not you had a chance to look at modern BERT in addition to the sort of older BERT family of models. It has larger context window and a whole bunch of architectural changes, but it's still sort of the BERT thing.
Okay. Thank you very much. Yes, I appreciate that a lot. So, as I said, we're not AI specialists. We are translational scientists. So we're trying to connect the dots here between different disciplines. This is very helpful.
I'm a theoretical chemist, but I haven't had a chance to actually play with modern BERT, but it's on my to-do list. I was just curious whether or not you could save me a trip to Hugging Face. So.
Well, that's please don't hesitate to reach out by email, and I will do the same actually, because if you're a computational chemist, then we do a lot of protein chemistry,
Yeah.
and some of this AI is actually being used for to predict
It's
docking and molecular
mainly the
dynamics
context
results.
window. So modern BERT has, so I think the original BERT family has something like, 512 quote unquote tokens, right? And I think that modern BERT context windows have much larger,
Okay.
or modern BERT has much larger context windows, which might let you do some interesting things with it. But
Okay.
it's still a small enough model, and it's based off the BERT architecture, right? Which you already are benchmarking against. So it makes sense that you can actually say something apples to apples versus
Mhm.
bringing in a GPT or some other crazy thing, right? So.
Exactly. Thank you so much. I really
Yep.
appreciate that.
Thanks so much, Gordon.